Welcome dear students. In this video we are going to learn about another G-protein coupled receptor signaling pathway. I have already discussed about two pathways of G-protein coupled receptor like muscarinic acetylcholine receptor pathway and rhodopsin that is present in our rod cells, and muscarinic acetylcholine receptor pathway is present in our heart muscle pacemaker cells. So these are all modes of G-protein coupled receptor. There are four type of, or you can say 5 type of modes of this G-protein coupled receptor. In this class we are going to learn about how G-protein coupled receptor signaling pathway activates another effector molecules called adenylyl cyclase, then cAMP, then PKA. What happens here, let's suppose this is a plasma membrane and this is the G-protein coupled receptor. Let's suppose this is a GPCR. This adenylyl cyclase is an enzyme associated with the plasma membrane, can be activated by some hormones like glucagon hormone or ACTH, that means adrenocorticotropic hormone, and also adrenaline hormone, or you can say beta adrenergic hormone, adrenaline hormone. These three hormones, when binds with a G-protein coupled receptor that is associated with a G-protein called G-alpha, G-protein means trimeric G-protein that has alpha, beta, gamma. The alpha subunit of the G-protein is actually a stimulatory protein. You have to remember this. I have mentioned here that in case of muscarinic acid, the G-alpha protein is inhibitory, I. Okay, here, and in case of rhodopsin, the G-alpha protein called G-alpha T, T for transducins. So each and every GPCR pathway, the G-alpha protein is different. Here the G-alpha protein is S. S means stimulatory. That can activate a protein that is actually enzyme. Let's suppose this is the protein and we are calling it adenylyl cyclase enzyme. What does it mean? That means it can convert ATP, adenylyl, that means ATP, cyclase means is cyclic form. So this enzyme can convert ATP to cAMP. The cAMP is a second messenger. This is a second messenger. Okay. Now how this adenylyl cyclase is activated? When glucagon, adrenocorticotropic hormone and adrenaline, this hormone sit on this G-protein coupled receptor, this G-protein coupled receptor acts as a GEF. I have mentioned in my previous class, GEF means guanine nucleotide exchange factor. That means this G-alpha S protein at first is associated with a GDP, and in that form it is actually off in state, off. But when these hormones bind with the receptor, this alpha protein actually exchanges GDP with GTP and becomes on. In that conformations, this G-alpha protein can activate the adenylyl cyclase enzyme. At first it is inactive in conformation, then it activates the adenylyl cyclase enzyme. Then ATP, from the ATP cAMP actually produced, and this acts as a second messenger. How it actually works here? Yes, another thing I want to mention here, that there are some other G-protein coupled receptors are there in the same cell. In the same cell, there is the other GPCRs are there that can receive another signaling from different hormones like prostaglandin E1 and adenosine. Adenosine is a inhibitory molecule. Okay, so these two molecule, when bind with a particular G-protein coupled receptor that is associated with another G-protein G-alpha I have mentioned here, this is G-alpha inhibitory, that when these molecules bind with GPCR, it activates this GPCR activate this G-alpha I into GTP conformation. Then this molecule G-protein actually perform the opposite function of G-alpha S, inhibit the adenylyl cyclase. Now let's suppose this process is not occurring, this glucagon or adrenaline hormones are actually secreting. So they bind and they activate the cAMP. This acts as a second messenger. How this cAMP activate this PKA? There is a very actually intricate molecular mechanism. The PKA, that means protein kinase A, has 4 subunits. One is called there are 4 subunits of this PKA. This is a total PKA. These molecules are called regulatory subunit, and this green are called catalytic subunit. That means this portion, this green portion, is actually the active enzyme, but it is actually suppressed by the regulatory subunit of the same protein. Now, when cyclic AMP, there is binding site in the R or regulatory region. Two cAMP can bind with each R subunit. So, if cyclic AMP binds with this regulatory subunit, then a structural changes occur in the R or regulatory subunit of the PKA, that they no longer can bind with C or catalytic subunit. So, the catalytic gets region. In this case, I want to mention that the binding of cAMP is actually cooperative in manner. That means when a one cAMP binds with the single R or regulatory subunit, it actually helps the binding of other cAMP with more actually spontaneously. That is called cooperative binding. That means when one cAMP binds with the first regulatory region, it actually changes the R or regulatory subunit in such a way that it can receive the second cAMP with high affinity or actually spontaneously. So it's called cooperative manner. So cAMP binds with the R subunit cooperative manner. When it binds with the R subunits, it changes conformation. It no longer can bind with the C catalytic subunit. So the catalytic subunit becomes active. Okay, actually open and becomes active. Now this catalytic subunit of the PKA function, how it functions, I will show you. What happens? Here I have mentioned 3 hormones. Specifically, you can remember 2 hormones, that is I have mentioned, that is glucagon and adrenaline hormone. These 2 hormones actually works in 2 particular cells we call muscle and liver cells. So actually on these 2 cells, muscle cell and liver means hepatocyte cells. In case of glucagon, the name implies that this hormone actually interact or oppositely interact with insulin. Glucagon helps in 2 important things, that is glycogenolysis and glucogenesis. There are 3 terms actually there. These 2 hormones helps in these 2 processes. Another term is called glycogenesis. What these 3 process means? Glycogenolysis means breakdown of glycogen. Neoglucogenesis means production of glucose from other 3 carbon molecules, and glycogenesis means production of glycogen from the glucose. So these 2 events are actually opposite to the third event. Here glycogen is produced. Glycogenesis means production of glycogen, and in this case glycogenolysis, that means production of glucose, and also in this case production of glucose. So this 2 event is actually opposite to this glycogenesis. This 2 hormone and ACTH hormone also helps in this 2 process and inhibit this glycogenesis, so that glycogen is not produced, only glucose is produced, because when there is need of glucose, then glucagon and adrenaline hormone actually secrete out, and that actually helps to produce. And how it occurs, I will show you the mechanism, how PKA actually work in this way. Because now this PKA has 2 catalytic subunits. In the cell of muscle or liver, there are 3 enzymes are actually present. that is called GPK, that means glycogen phosphorylase kinase. Glycogen phosphorylase kinase. This is an enzyme, this is a kinase enzyme. This is actually inactive in conditions, inactive, or I can say off, remains off in condition when there is less amount of this hormone. When the hormone is less, then the cAMP, that means cAMP level is also low. When cAMP level is low, this hormone is off in state. Now when the cAMP level rises, because the hormone binds to the cells, this catalytic subunit becomes active. This catalytic subunit of the PKA, protein kinase A, that means it is a kinase, can phosphorylate this GPK and can activate it, so it becomes active. Now active GPK can phosphorylate because it is called glycogen phosphorylase kinase. It is also a kinase. This protein phosphorylate another protein called GP. This is also off in state. But when this GPK phosphorylate, active GPK phosphorylate GP, that means glycogen phosphorylate, it becomes active. When GP becomes active, it actually helps in the breakdown of glycogen to glucose 1-phosphate. Okay, so GP actually helps in the breakdown of glycogen. This is called glycogenolysis. Now this glucose 1-phosphate is actually converted into glucose in case of liver and actually export out into the blood by a transporter called GLUT2. And in case of muscle, this glucose 1-phosphate is actually converted into glucose 6-phosphate and actually go to the TCA cycle. So it produces energy. So here glycogenolysis, that means production of glucose. But here another thing is there, an enzyme called glycogen synthase that helps in the glycogenesis. Glycogen synthase that helps in the synthesis of glycogen. This is actually on in state. This enzyme is on in state. Now this enzyme should be off. Then this PKA phosphorylate this GS. This means glycogen synthase, and becomes the GS become off. So glycogenesis is inhibited, but glycogenolysis is actually promoted. Another thing is neoglucogenesis. That means production of glucose from other 3 carbon molecules like pyruvate, lactate. How it occurs? There is another mechanism I will show you. That is, this catalytic subunit of PKA, catalytic subunits of PKA can enter into the nucleus. And let's suppose this is a gene. This is the gene. Okay. Here I want to mention that the hormones that actually activate this adenylyl cyclase and activate or increase the concentration of cAMP. This cAMP activates PK. Now there is a gene that have a particular site actually called, that is called cAMP response element. In short, we call it CRE. This is a DNA region. This region. Let's suppose this region is the CRE region. That means this region is actually sensor of the cAMP. Now there is a protein called CRE binding protein. CRE binding protein. CRE binding protein. In short, we call CREBP binding protein. CREBP. This protein can bind with this area. So let's suppose this is the CREBP. This is the CREBP. This green color protein is the CREBP. But this protein present in the nucleus but inactive in state. When protein kinase gets activated, this catalytic subunits enter into the nucleus. Some catalytic subunit enters into the nucleus and phosphorylate this CREB protein. When it gets phosphorylated, the CREB proteins associate with another protein called coactivator. Its coactivator name is CBP P300. This is a very important coactivator. CBP-P300. This co-activator actually touch or actually get connected with another protein called RNA pol2. Now you can see pol2, that means this gene can be transcribed. So not only CREP actually bind, there is a co-activator that actually connect with the RNA pol2, then the gene actually produced, and the gene actually that helps in the neoglucogenesis. Okay, so this is all about the third mechanism. So in this video we have completed the understanding of this third mechanism. Then in my subsequent classes I will talk about how PLC beta is activated and PKC gets activated, and NO, that means nitrogen oxide is produced. So hope this video will help you. If you have any doubt about this topic, you can write me in the comment section. Thank you for watching, and always keep learning.